Hey everybody, it's Damon Tighe from Bio-Rad Laboratories. I'm here to show you how to adapt two of our existing kits to fit within a COVID-19 context. We're going to play with the Science of Opioid Dependence kit, which is a great lab for teaching hands-on gel electrophoresis and having the ability to teach students about the theory of PCR, the polymerase chain reaction. We'll also look at how to place the ELISA Immuno-Explorer kit within a COVID-19 context. But before we do that, let me give you a brief background on Bio-Rad and myself so that way you know where these tools are coming from and where any biases might be. Bio-Rad Laboratories, if you're not familiar with them, is a biotechnology company that's been around since the 1950s. They were started near kind of UC Berkeley by a husband and wife, and they've grown this business to be a 7,000 plus person business that operates worldwide. Life science researchers use our tools to do their research and produce their work products. Pharmaceutical companies use our tools to produce their drugs. And our clinical diagnostic kits end up in hospitals all over the world. Bio-Rad has an education unit due to a high school teacher joining Bio-Rad a little over 2 decades ago. When he joined, he asked the CEO at the time, hey, how can I get what's going on at the leading edge of biotech into classrooms? And in a sustainable way, because I was basically dumpster diving, asking for handouts, building curriculum around that, and then when those things ran out, I'd have to start the whole process over again. So I want to make something sustainable. The CEO said, you know what, I'm not quite sure how to do that. I'm going to give you a little bit of funds, a little bit of responsibility. Let's see what you do. What Ron did was he built the Bio-Rad Explorer program, which more or less takes these biotechnology techniques and translates them into kits that any teacher could pick up and run in their classroom. The beauty of this is it relies upon the industry quality standards of Bio-Rad, so these things work. And you get all of the technical support and resource support that Bio-Rad provides to its normal customers. So you can call our tech support line, talk to people right away about any troubleshooting needs you might have on a kit. You can contact curriculum and train specialists like me who've had a background in the classroom, usually some research. We can help you also troubleshoot if you need it, or figure out how to modify a kit to fit these curriculum needs that you need to hit within your classroom. My background is I came from the national labs to Bio-Rad. I used to work at the Joint Genome Institute. I worked on the Human Genome Project. And then prior to that, I was a high school teacher in Portland, Oregon. The main reason I joined Bio-Rad was my experience at the national labs, hiring people into the laboratory. I found that we could get people that were coming out of four-year universities that had great theoretical knowledge, but they didn't have those hands-on skills. As an employer, I'm hiring you for both of those things. And so I joined Bio-Rad to help drive more of this hands-on, skills-oriented biotech education lower and lower in the education pipeline, because having skills for a student early on opens up all kinds of doors, jobs and within the educational sphere. And so without further ado, let's jump into adapting these two labs for a COVID-19 context. This slide deck is going to allow us to adapt Bio-Rad's Science of Opioid Dependence kit into a COVID-19 context for student engagement. You can find this slide deck on biorad.com/teachingcovid, where you'll also find other resources like slide decks on the biology of SARS-CoV-2, slide decks on detection methods for SARS-CoV-2, and a number of hands-on technique-driven labs that are put within a COVID-19 context. This specific slide deck is broken down into 3 sections. First, kind of getting started, the introduction that you need in order to kind of understand where this is going, where some of these additional resources are, and then instructor preparation, which is basically how to kind of take what's given to you in the Science of Opioid kit and tweak that so that it perfectly fits this COVID-19 context. And then a set of student slides that allow you to basically execute this lab with students, that gives them the scenario and all the protocol steps that they need in order to run this successfully. The background on this lab is that we tried to set it within a historical case study. And so this lab is going to place students basically as a group of researchers looking at this famous case study from early 2020, where you had 3 families that went to a small restaurant in Guangzhou, China, and slowly they started coming down with the symptoms of COVID-19, and subsequent testing was done to then figure out who was infected at the restaurant. The thing to remember is this is really early on in the pandemic. People were still unsure of how the virus transmits. And so this lab experience is to place students in that position where they're going to use the evidence that they derive from running gel electrophoresis, which is going to tell them which of these patients have SARS-CoV-2 and which ones don't. They're going to use that with a map of the restaurant to then come up with hypotheses or claims of how this virus is being transmitted. The goal isn't to point them to one right answer. The big goal is actually to get them to come up with multiple claims, use the evidence that they've built through gel electrophoresis and combining their data with everybody else in the class to then be able to reason which one of these claims holds the most water. One thing to keep in mind here is that we are using an existing kit with a new context. So the baseline kit is the Bio-Rad Science of Opioid Dependence kit, which has students use gel electrophoresis to analyze whether or not there is genetic correlation to opioid dependence. So we're taking basically the reagents from that kit and then recontextualizing them so that students can evaluate it within this COVID-19 pandemic. One thing to note there is that it is still useful to download the Science of Opioid Dependence Kit instructor manual from the website, especially for protocols on how to make your running buffer, your gels, and things like that. Once again, that biorad.com teaching COVID website has those additional SARS-CoV-2 biology and detection slide decks, and there's also links out to other useful content, especially in the context of this lab where students are going to be running gel electrophoresis of samples that have gone through the polymerase chain reaction. So that website also has links out to our interactive animation around PCR, the polymerase chain reaction, and links out to the Explore YouTube channel where there are technique videos for just about any technique that you're doing in your classroom. Everything from using a transfer pipette all the way out to doing a Western blot. And in this case, there's a series of videos that show students the techniques of how to pour gels, how to load gels, how to run gels. The other thing to be aware of for these videos is these are really, really useful tools as far as pre-labs go, having students watch these before they come into the classroom to then execute that technique. And within distance learning, these can be used as substitution for when students can't be there in person to do these techniques. All right, we're jumping into the student portion of the slide deck, so I want you guys to put on your student hats and think about this as a student would. So the scenario in our class is that we've got an individual that ate dinner at a restaurant in Guangzhou, and later that evening that individual experienced some fever and some cough, and they went to the hospital. The hospital quickly suspected that this might be a COVID-19 incident. And so they immediately got the rest of the patrons from that restaurant, one waitstaff and one cook, and put them on a 14-day quarantine. Nasopharyngeal swabs were collected from all parties involved. What is COVID-19? Well, actually, we should probably break down this nomenclature before we move any further. So, coronavirus. A coronavirus is a viral particle that has a corona if you look at them underneath an electron micrograph. And what this means is it has this crown on the outside that's caused by the spike proteins. Coronaviruses is a family of viruses that can infect humans and animals. They circulate widely within human populations, but some strains can be very, very bad for us. SARS-CoV-2 is the Severe Acute Respiratory Syndrome Coronavirus 2, which is the strain causing the current pandemic. The reason it has the suffix 2 on it is because there was a former severe acute respiratory syndrome that happened in the early 2000s that also emerged out of China. There was also another near-pandemic coronavirus called MERS that then came on the scene in 2014, 2015. COVID-19, though, the coronavirus disease 2019, describes the set of symptoms caused by the SARS-CoV-2 virus. The structure of SARS-CoV-2 is something that we have to understand in order to understand how testing is taking place with this virus. So the first thing to look at is that this is a large RNA virus. Right there in the center, that nucleic acid material you see, that is a positive strand RNA genome. On the outside of this virus, we have those large spike glycoproteins. In the case of SARS-CoV-2, these interact with ACE2 receptors, and this is how they gain access to our cells. The SARS-CoV-2 genome is huge. It's almost 30,000 nucleotides, which is very, very large for an RNA virus. And this is actually great for us because this means it has a proofreading enzyme in there, which minimizes the mutation rate. If we want to figure out if those patients from the restaurant have active SARS-CoV-2 infections, this is the generalized workflow that we would use. We would collect nasopharyngeal swabs from them, we would extract RNA from those samples, use reverse transcription to take the RNA, turn it into DNA, and with that DNA now we can perform PCR, an amplification technology that can use specific primers that tell us whether or not, say, the virus was there or whether we got human DNA out of those extractions, and then use this to perform analysis to determine who was infected and who was not infected. The polymerase chain reaction, also known as PCR, is a process that allows us to amplify, say, one copy of DNA into hundreds of millions of copies of DNA in just a couple hours, which gives us enough DNA that we can then put in an agarose gel and be able to see with our eyes. PCR uses primers, small pieces of DNA that are complementary to regions in our target sequences, in order to isolate and amplify just small sections of the nucleic acids that are in a sample. It does this using a polymerase, the enzyme that actually makes copies of DNA, and by modulating temperatures, which can allow DNA to unwind, for primers to bind, and for that polymerase to become active. PCR can then reveal if a certain DNA sequence was in a target. So in our case, we're going to use PCR to reveal whether or not that SARS-CoV-2 virus was in our samples. All right, now that we've got the theory under our belt on how some of these techniques work, let's rejoin our scenario. So our class has received the patient samples from the clinical diagnostic lab that we're working with. They've already done the RNA extraction, the purification, the reverse transcription, and they've applied what we call a multiplex PCR to the samples. This basically means they're using two separate primer sequences to go after different targets. The first target is a 530 base pair sequence coming out of the SARS-CoV-2 end protein coding area. The second is a 250 base pair region that's coming out of the human genome in the GAP-DH gene. This is going to act as an internal control that verifies that all those previous steps, the RNA extraction, etc., worked out all right. Our task is going to be using gel electrophoresis now on those multiplexed PCR samples to determine which patients were infected with SARS-CoV-2. We're then going to pool that patient data together and propose ideas of how that virus may have transmitted to the different patients. We're going to do that by looking at this schematic of the restaurant and tracing back which patients are infected and where their position was in the restaurant. Reverse transcription PCR is simply where RNA is isolated from the sample. That RNA then is run through reverse transcription to turn it into DNA, and that DNA then can feed directly into a PCR amplification process, generating you many copies of your target DNA that originated in, say, your viral sample. Gel electrophoresis is the technique that we're going to deploy in our lab, in order to figure out which samples have amplified. DNA has an overall negative charge, which we are going to exploit because if we put it inside of a circuit, i.e. a horizontal gel box, we can take that negative charge and get it to migrate towards the positive side of the chamber. If we put a sieve in front of those DNA samples, they will separate out based upon their size as they migrate their way through the pores in the gel. So before cutting you loose in the lab to run those samples, I want you to actually make predictions about what that gel is going to look like. So on this gel image here, what I'd like you to do is take these samples and illustrate them on the gel. So you're going to have a molecular weight marker, which is going to have 1,000 base pair, 700, 500, and 200 base pair bands. You'll have a GAP-DH positive control. Remember, GAP-DH should be about a 250 base pair band. You'll have a SARS-CoV-2 and GAP-DH positive control, so we're looking at a 530 and a 250 band. You'll have a negative control, and then you'll have a patient sample that's positive for SARS-CoV-2 and a patient sample that's negative for SARS-CoV-2. So I want you guys to just go ahead and sketch those in on the gel that you see there at the right. Once you've done that, we can now cut you loose to the lab to run the protocol. So what you'll find at your lab bench is that you'll have molecular weight marker that you're going to drop in lane 1 of your gel. You're going to have GAP-DH positive control in lane 2. You'll have SARS-CoV-2 and GAP-DH positive controls in lane 3. You'll have a negative control in lane 4. And then you're going to have 4 patient samples based upon the letters and numbers that are on the respective tubes at your desk. You'll be running these in a 1% TAE agarose gel in a horizontal gel chamber. So I'll have you guys basically fill the electrophoresis chamber with enough TAE to cover the gel by about 2 millimeters. You'll then use a fresh pipette tip each time to load 10 microliters of each sample into each one of the wells as described, and 20 microliters of that molecular weight ruler into lane number 1. Place the lid on the electrophoresis chamber, clip in the leads, and turn the power on as your instructor tells you which voltage and time increments to use to run the gel appropriately. Once the gel has finished running, you'll go ahead and shut down the power supply and carefully remove the gel in order to do imaging. If you're using a UV stain, such as the UView, you'll be able to take your gel and go right onto a UV transilluminator to look at your bands. And if you're using Fast Blast, you're going to take your gels and go into a staining tray with Fast Blast. Now that you've stained and visualized your gels, I'll have you guys analyze which patients were infected with SARS-CoV-2 from the restaurant. You're going to take your data and combine that with all of the other student work groups' data onto the restaurant seating chart to give us a visualization of where infected people were in the restaurant. Now, I'm kind of jumping out of the student slide deck right now and going into some of the instructor prep just so that you guys can see what some of these band results look like. So on the left there is a molecular weight marker, and on the right is a patient that's positive for SARS-CoV-2 and then one that is negative. Note that both of them are showing GAP-DH, which tells us that in both cases we had successful extraction of nucleic acid and downstream processing of the patient samples. The students are going to be plugging into this chart here, basically filling in circles red for those that are infected and blue for those that are uninfected. So ultimately what you'll get is something that looks similar to this. This might vary a little bit depending upon student success with gel electrophoresis, especially if it's their first time. But with this now, students can begin to formulate hypotheses on how this virus is actually being transmitted. Once students have that full data picture of what was going on in the restaurant, I would send them back to their groups and have them tackle these 3 prompts. So we know that patient B3 was the original customer to report symptoms and visit the hospital. So using that class data, Propose a possible explanation of how the virus spread at the restaurant. And then next, propose a second alternative explanation for how the virus could have spread. What additional information and/or data would help you determine which proposal is more likely? The reason to run students through multiple potential ways that the virus could have spread is because this is often what has to take place when you have an emerging virus. You need to look to see what are the possibilities and then start trimming those down based upon the data that's there. So depending upon your student group, you might need to help them kind of move in the direction of more than one potential hypothesis. So some hypotheses that students will come up with are things like food or utensils were contaminated. You might have infection that took before or after the meal. Say there was a common wait space at the restaurant and certain groups of people were all waiting together. They could have contaminated there. Or maybe there was an elevator ride that you needed to take to get to the restaurant. Some people took the elevator, some took the stairs. There could have been other various transmission events unrelated to the restaurant that befell the different families that showed up. It could also be that you have some sort of air droplet movement of the virus from one table to the other, especially if you take a look at where that air conditioner unit is. But the big picture here is to get those students to propose multiple ideas and then start trimming them down based upon the available data. And if there isn't enough data to support their hypothesis, having them state what data they would need to see then in order to determine whether that hypothesis is plausible or not. All right, now that you've seen the meat of the lab, let me show you what you need in order to run this. Remember, we are adapting this from the Science of Opioid Dependence kit, and so what you'll need is you'll need to buy one of the two flavors it comes in, either the Science of Opioid Dependence kit with the Fast Blast dye or the Science of Opioid Dependence kit with the UView electrophoresis reagent pack. The latter, which is a UV fluorescent stain that Bio-Rad uses in its kits. Those kits come with all the reagents that you need in order to run the lab. You do need some hardware to run this lab, so you need a 20 to 200 microliter adjustable micropipette for the instructor in order to do aliquoting with. The students will need 2 to 20 microliter pipettes or 10 microliter fixed volume pipettes in order to load the gels with. They'll need gel electrophoresis boxes with power supplies, marking pens, calculators, and a UV transilluminator if you're going to use the UView kit, or gel staining trays if you're going to use the Fast Blast version of the kit. So let's jump right into this slide deck that helps you take ELISA's Immuno-Explorer kit and put it within a SARS-CoV-2 COVID-19 context. This slide deck, when you download it, has 3 sections to it. A getting started that has the introduction and kind of background on SARS-CoV-2 and the ELISA kit, instructor preparation, and then student slides. For this video though, I'm going to invert these a little bit and I'm going to do the student slides before the instructor preparation, just so that way we can get right into the meat of the activity and you can see that first. Bio-Rad's ELISA Immuno-Explorer kit allows you to explore antibodies and how they're used to detect diseases with your students. The kit uses real antibodies and real antigens. However though, these are a simulation for COVID-19. These are not human samples, and these are not human antibodies that are being used. If you go to the website for the ELISA Immuno-Explorer kit, you can download the instructor manual there, which brings you through the different ways that you can use the kit in different contexts and to show different types of ELISAs. So you can go through antibody detection, you could go through antigen detection. On the product website, there's also 2 great resources to know about that can really enhance how well students pick up the theory of ELISA. The first is an interactive animation that students can use to step their way through an antigen detection ELISA. The second is an ELISA paper modeling activity, which helps students kind of kinetically build their way through how an ELISA works. I personally actually like to use these simultaneously, so I'll step through the interactive animation while participants use those modeling pieces to build what they see in the animation at their desk. Sometimes I'll take it even a step further, and on the back of those modeling pieces I'll put instructions about what you would do at that step, i.e. how much material you would add to a well and how long you'd let it incubate. So that way, at the end of running the animation and doing this paper modeling, they can literally flip their paper models over and they have a protocol which they can apply to the ELISA that we then run in class. What I really like about this lab is that everything you need actually comes in the box. You even have enough transfer pipettes to run this kit with those. However, though, if you can use real micropipetters, you'll be able to teach real skills to your students. If you don't have the capital for the micropipettes, you can always start by using those fixed volume micropipettes that are significantly cheaper. They just do one volume, but they're kind of the in-between the transfer pipette and an adjustable micropipette. So the scenario your students are going to be working in is that they are in a diagnostic lab that's using an ELISA antibody test to determine if patients have been exposed to SARS-CoV-2 or not. When patients are infected, of course, with SARS-CoV-2, their immune system creates antibodies in response to the virus. If you do a blood draw and then pull serum from that, you can use that in an ELISA antibody test to figure out whether or not that patient had previously been exposed to SARS-CoV-2. In general, your student stations are going to look like this. You'll have some wash buffer there in a container. You'll have the reagents necessary for the ELISA. That's going to be a positive control, a negative control, antigen, secondary antibody, substrate, and then those yellow tubes are going to be the patient samples. You'll have a microtiter plate, which is where all of these reactions are going to take place. You'll have at least one transfer pipette that's going to be used for the wash buffer, and you could have other transfer pipettes there in order to use for the other reagents, or you could have a micropipetter or a fixed volume pipetter and a set of tips. A Sharpie will be helpful for labeling the wells, and a stack of paper towels will be helpful because we'll be using that as a place to deposit liquids as they come out of the microtiter plate. All right, grab that Sharpie and let's label up that microtiter strip. So what you're going to do is you're going to put your controls on the left-hand side, your patient samples on the right-hand side. We're going to do 3 replicates of each, which is essential in this lab just because we're having students micropipette things, and so there's the potential for cross-contamination. Having 3 replicates is great because what will take place is, usually if an error took place, you'll get contamination in the first well, and the last 2 wells will be representative of what was supposed to be going on in that well. Now that your microtiter plate is labeled, we can get to adding our first reagent to the plate. What we're going to do is we're going to take the green tube that says AG on it for antigen, and we're going to add that to every single well in this strip. If you're using a transfer pipette, you can literally just add a drop to the bottom of every single one of those wells. If you're using a micropipette or a fixed volume pipette, you're going to use 50 microliters of antigen for every single well. After we've deposited them, we're going to wait 2 to 5 minutes for these antigens to actually bind to the plate. But first, we should probably talk about what is an antigen. Antigens are just foreign substances that trigger an immune response within the body. The CDC recommends using the SARS-CoV-2 spike protein as the antigen for an antibody ELISA. Antigen binding takes place on these plates because the plate is polystyrene, and proteins have these hydrophobic regions to them. And when they're in an aqueous solution, they will usually try to crowd up against other hydrophobic regions. And so they can do this with that hydrophobic wall, basically, of the polystyrene plate. And so the antigens will literally kind of stick themselves to the plate because of their fear of water. But why add an antigen to the wells first in the case of this ELISA? Well, the reason we're going to do that is because ultimately we're going to be looking through the serum to see if we can find a primary antibody that's going to target our antigen. And our antigen in this case is, of course, the spike protein from the SARS-CoV-2 virus. Now that our 5-minute incubation is up and that antigen is well bound to the polystyrene plate, we are going to wash out anything that's not stuck to the plate. So we're going to grab a fresh transfer pipette. We'll label it with our Sharpie wash so that we can continue to use this transfer pipette for all the subsequent washes going forward. And what we'll do is we'll take a big pull of the wash buffer, and then we'll go through and we'll add a little bit of wash buffer to each one of the wells. In this case, I would say fill those wells about 3/4 the way up. And then once they're full, what we're going to do is we're going to invert it and then tap it out onto the paper towels at your desk. So you're going to do this once, and then you'll do it again. Why is washing important? Well, it's going to remove any of those unbound antigens, and then it also is going to block the rest of the well surfaces so that nothing else binds specifically. We'll get more into this when we talk about the prep at the end, but the wash buffer is basically PBS, which is the generalized carrying solution for most proteins, and a little bit of surfactant, tween 20, which is what's going to coat the surface of those plate wells and make it so that no new proteins can bind to the polystyrene. Once you finish tapping out your second wash, we can move on to adding our controls and patient samples. Just like before, if you're using a transfer pipette, you're just going to put a drop in the bottom of each one of these wells. And if you're using a micropipette or a fixed volume pipette, you're going to add 50 microliters to the bottom of each well. It's important to switch tips in between each tube or switch transfer pipettes between each tube to get rid of the chance of cross-contamination. So for the first 3 wells, we're going to add our positive control, basically our tube containing antibodies. So we're going to grab our violet tube and we'll add basically sample to those first 3 wells. To the next 3 wells, we're going to add our negative control, which is going to come from our blue tube. And then to the next 3 will be our first patient sample, and then to the last 3 will be our last patient samples. These will also incubate for 2 to 5 minutes to give time for the antibodies to attach to the antigens that are bound to the plate. Antibodies are the key constituent of ELISAs. And this is because of their ability to detect very specific antigens. So one antibody will detect one specific antigen. If we look at how an antibody is laid out, they're kind of a Y shape. We've got a constant region in kind of the stem portion, and then those outer tips of the Y is where the action is taking place. So changes in protein codes there dictate what antigens it will bind to. In the human, we have almost 3.5 million different antibody combos potentially floating around in us, which is pretty amazing considering the fact that we only have about low 20,000 genes if you look at the human genome. So how do we get that immense diversity of antibodies? It's through variable splicing events. So within the genome, we've got 65 V regions, 27 D regions, 6 J regions, and then 3 constant regions. And the reshuffling of these gives us that great diversity, which then means we can recognize literally millions of different things that might show up in our bloodstream, be able to attack them with an antibody, and then that constant region can kind of harness the rest of the immune system into that area to then fight whatever that foreign body is. And after filling you in on how antibodies work, we've pretty much blown the amount of time that we need for those primary antibodies to stick to the antigens that are in the plate. And so this is a technique that you can use with students if you're not basically dropping them the protocol and then going off and doing something else. You can help lead them through the procedure and then fill in these time gaps with more details about what's taking place at each stage. But what we need to do now is we need to wash these wells out. So I want you to grab that transfer pipette with the wash buffer in it and go ahead and fill those wells near full and then tap them out. And then we're going to do this one more time. Fill those wells near full with wash buffer and then tap them out once again onto your napkins. Some of you may be disturbed that there is a little bit of foam residue in the bottom of your wells after tapping them out. You don't need to worry about that because every time we do an addition of a reagent, it will sink to the bottom underneath those bubbles, and so those bubbles really don't matter. So the next thing we're going to do with this plate is we're now going to add our secondary antibodies, which are going to basically probe all of those wells looking for that primary antibody. So if you're using a transfer pipette, you'll just add a drop of secondary antibody to each one of the wells. If you're using a fixed volume pipette or a micropipette, you're going to add 50 microliters of secondary antibody to each of the wells. This is going to be another 2 to 5 minute incubation period Anytime you can go the full 5 minutes, you'll get a much stronger reaction, but if you're squeezed on time in your classroom, you can usually get away with 2 minutes and you'll just get a little bit slower reaction at the end, but they will still work. So those secondary antibodies that we just added to the plate, what they're going to do is they're going to bind to the constant domain in those serum antibodies that have potentially bound to antigens that were bound to the plate. These secondary antibodies kind of do two things for us. A, they've got an enzyme linked to them, which is going to ultimately allow us to get a colorimetric output from this. And B, multiple secondary antibodies can bind to one serum antibody, which means that we can actually amplify the signal from one serum antibody into multiple enzymes being there, which means that this reaction can take much, much faster. So instead of waiting an hour for these results, you'll be able to get these results within minutes due to that signal amplification. Once that 2 to 5 minute incubation is up, we're going to wash our wells again. However, though, this is going to be a little bit different than the last 2 wash cycles, because instead of running 2 washes, we're actually going to run 3, and that's mainly so that we can clear out all of that secondary antibody, because since it's got an enzyme there, we don't want any residual enzyme being left around, otherwise it could lead to false negatives. So just like before, you're going to fill up your transfer pipette with wash buffer, you're going to fill those wells near full, and then you're going to tap those out onto your napkins, and you'll do that for 3 rounds. All right, now that you've knocked out your 3rd wash, what we can do is we can add our final reagent to the plate. We're going to add 1 drop of substrate to every single one of those wells if you're using a transfer pipette, and then 50 microliters to each one of those wells if you're using a micropipette or a fixed volume pipette. We're going to wait 2 to 5 minutes to get this reaction to take place, but usually this takes place way before 5 minutes. And what we'll see is we'll see a color change in wells where secondary antibodies were present, because those secondary antibodies have the enzyme that can break down this substrate, causing a blue product. By the end of the 5 minute incubation, your plate would look probably similar to this, where your positive control, those first 3 wells, will be this nice blue color. Your negative controls will be clear, and then, depending upon the status of your patients, they'll either be blue or clear. So those blue ones, of course, mean that that patient had SARS-CoV-2 because they had serum antibodies that bound to the antigen, a secondary antibody bound to that, and then the substrate was being processed by the enzyme on the secondary antibody. Now that you've been through what the students would see, let me back up and we'll do how to adapt this from the original ELISA, and then also show you a little bit about the prep. So the original manual for the ELISA Immuno Explorer Kit comes anytime that you order the full kit, and you can also download it for free from the product webpage. The way to adapt it is basically by using this little chart beneath and following the ELISA Immuno Explorer Kit Protocol 3, the antibody test, in order to do the prep of the reagents. This series of slides is going to step you through visually how you prep this lab. So the first thing you need to do is make 1x PBS solution. PBS just stands for the phosphate buffer system, which is a general buffering system for when you use proteins. It helps them maintain their structure. So you're going to take 10 milliliters of the 10x PBS that comes in the kit, and you'll add that to 9 milliliters of distilled water. The next thing you're going to do is prepare wash buffer. Wash buffer is just going to be 805 milliliters of distilled water plus 90 mils of that 1x PBS that you just made, and 4.5 milliliters of the Tween 20 that comes in that little small bottle in the kit. I personally like to mix this into a jar just so that it's easy for me to pour wash buffer stations out for the student stations. You should notice that after mixing it, it has a little froth at the top, and this is from the Tween 20. Tween 20 is a surfactant that's often used to solubilize proteins. In this case, it will help us solubilize proteins, and it also helps us coat the polystyrene plate so that no additional proteins can bind during our ELISA after our original antigens have bound. Our next step is going to be making our 50x stock solutions from our lyophilized primary antibody, secondary antibody, and antigen. These all come in glass vials with those metal tab crimped lids on them. And so a lot of times you can get those to pull off cleanly, but if you can't, I would recommend bringing a ruler to help kind of pry those metal lids off. Once those are off, what you're going to do is you're going to use a fresh pipette tip every time to add 0.5 milliliters of PBS to each one of those vials. I would then put the lid back on, give them a good shake, and then set them on the bench for a couple of minutes before I go to work with them further, just to make sure that those proteins fully dissolve into solution. The first working solution we're going to make is of our antigen. So we're going to grab a 50 ml tube, we'll label it antigen. We're going to add 24.5 milliliters of 1x PBS, and then we'll add 0.5 milliliters of that 50x antigen stock from the glass vial. Our second working solution that we'll make is our primary antibodies. We'll grab another 50 ml tube, we'll label it PA for primary antibody. We'll add 24.5 milliliters of wash buffer, and then add 0.5 milliliters of that 50x primary antibody stock solution. We're going to do a similar thing for the secondary antibody. We'll grab a 50 ml tube, label it SA for secondary antibody, add 24.5 milliliters of the wash buffer, and then add 0.5 milliliters of that 50x secondary antibody stock solution. Now that you've got those working stock solutions, we can now aliquot them into the tubes for students to use. So the first one we'll do is we'll grab our 12 violet microtest tubes, we'll label them positive, and we'll add half a milliliter of 1x primary antibody to all of those tubes. The next thing we'll do is we'll grab 12 blue microtest tubes, we'll label those negative, and we'll add half a milliliter of wash buffer to each one of those tubes. We'll then grab 12 green microtest tubes, label those AG, and aliquot out 1.5 milliliters of the 1x antigen to all of those. Then grab the orange microtest tubes, 12 of those, label those SA, aliquot out 1.5 milliliters of the 1x secondary antibody to all of those. Last one, we're going to grab 12 brown microtest tubes, label those sub for substrate, and aliquot out 1.5 milliliters of the HRP substrate into all of those tubes. And now for making the patient samples, you'll grab the baggie full of yellow microcentrifuge tubes. And what you want to do is count out as many as you have students. And traditionally what people do is they'll kind of split the tubes. 50% of these are going to be people that show up as infected, and then 50 are going to be people that don't show up as infected. So to half of those tubes, if you add 0.25 milliliters of 1x primary antibody, these will then be patients that will come up positive for SARS-CoV-2. And then to the rest of the tubes, you can add 0.25 mils of wash buffer, and those will all be patients that come up negative. You, of course, can mix up the percentages to play up different storylines and things like that if you're interested in being a little more creative with this. Once you've got all of these aliquoted, you can rack them up and then put them in your fridge at 4 degrees and hold on to them until the day that you go to do this lab. Bio-Rad's manual suggests that, especially with that secondary antibody being the most sensitive thing here, that only gets prepped out about 24 hours in advance. I've kind of played around with this quite a few times, and I would say you're actually really safe doing this, a number of days up in front. You just want to make sure that it stays cool because these are large antibodies, large proteins, and so thermal damage can take place, and then if contamination took place while you're pipetting, you don't want an organism in there eating your proteins up. You can then recover those tubes from the fridge for the day that you're doing the activity, and then rack them out into a student station similar to this, where you've got the patient samples, you've got the reagents that they need to run the ELISA, If they're using a micro pipetter or a fixed volume pipette, they've got tips to use, or they've got a whole bunch of those disposable transfer pipettes to use for every single step if you don't have one of those micro pipetting options. You'll also want a Sharpie there, and of course a stack of napkins for them to knock out their wash buffers onto.